Okay, so this is the last video for chapter 1 of part 1. Um, the same phenomenon that we finished with in the last slide that we were talking about actually may cause other alleles of the same gene to be expressed in instead. So that's a very interesting complication or implication of what we just talked about. So when we're talking about a recessive trait might actually appear instead of a dominant trait. So we're starting to understand that although when we break down genetics and kind of have it simplified, we're talking about only nucleotides in a certain sequence that's kind of easy to understand, and only about the nucleotides being read in triplets and so on and so forth. But actually, these two basic and simple mechanisms, when something goes wrong, may have severe implications to us. But at the end of the day, we're playing with nucleotides in a certain sequence, read in triplets that we call codons, that are, when we're talking about proteins, representing amino acids that are going to be connected, and other factors that we will learn about. But the idea is really, really simple, right? But it's also simple to have a mistake done, and then the outcome is not as we want it to be, for better or for worse. Okay, so when we have a mutation that is affecting the contiguous sequence of DNA, we call it a cis-acting mutation. All right? So the next question would be, all right, we understand that a gene also has regulating and recognizing factors. What happens if that is absent? So the gene theoretically is intact, and the information, the sequence that has a regulating or a recognizing role to play is absent. So in such a case, obviously, none of the alleles is going to be expressed, and then we call that mutation a trans-acting mutation, right? So cis-acting mutations act only on DNA, thus they will not appear in the RNAs or proteins, right? But trans-acting mutations can be found in multiple targets of the cell, right? So they can affect in more than one way the cell. So in conclusion, trans-acting mutations may occur in more variations and affect more processes and stages of the processes than cis-acting mutations. So the expression of genetic information usually works in one direction. It is said to be unidirectional, meaning going from DNA to RNA to mRNA, right, to protein production. We can find in nature, but not in all organisms, both single strand and double strand replication of DNA and RNA. So we have a term that's called retroviruses. And what they are able to do is to reverse transcription, meaning that they use reverse transcription to produce a single strand DNA or double-strand DNA. So reverse transcriptions makes transforming RNA into genetic information possible. So it's as if we're swimming upstream. These viruses are able to take an RNA and produce DNA from it. Okay? That means that reverse transcription opens a new possibility. Think about it. If there are organisms, be them viruses or other, it doesn't really matter, that can take RNA and produce DNA in that process that we call reverse transcription, it actually may be possible that RNA existed before DNA. So let's put things into order. In our body, the majority of processes go from DNA to RNA to mRNA to protein, right? But we have evidence in nature of organisms going from RNA to DNA, and that opens the possibility that maybe everything started that way and then it changed. Okay? And that means that it is possible that through reverse transcription, once again going from RNA to DNA, genes were actually inserted into the genome. Unbelievable. I mean, believable, but amazing. Okay, so you referred to a movie, and then we have our very last slide for this presentation that we show, and we know that in some viruses in nature, their genetic information cannot be duplicated by the organism itself. That means that that organism needs a host or host cells to do so. Okay, so that brings us to the end of the presentation of part 1, chapter 1. I hope you have enjoyed it, and once again, if you have any questions, feel free to communicate them to me.